During the 1930s, it was discovered that genes are located in the nucleus of an organism's cells, at specific locations on larger bodies called chromosomes. Each individual has a number of chromosomes which is standard for its species. In the case of humans, this number is 46, with 23 homologous pairs, having one from each parent. One pair of chromosomes is the sex chromosomes, in humans XX for females and XY for males. The remaining 22 pairs are known as autosomal chromosomes, and they have little to do with sex. The discovery of chromosome pairs provided a physical home for Mendel's paired genes. This means that a big Y, small yp will have its big Y gene on one homologous chromosome and the small y on the other corresponding chromosome. The number of chromosomes varies widely between species. Fruit flies of the genus Drosophila have 8. Dogs have 78, and some species of plants have over 250, like the mulberry with 308, having 22 copies of their 14 chromosomal units. Remember, a gene for a particular characteristic exists at a specific point on the chromosome called its locus, pronounced loci when plural. The purpose of the Human Genome Project was to identify the loci of all of the genes in our species. We may not know all the effects that a gene might have on the phenotype, but we now know where it is. Generally, a locus is home to one of two or more possible forms of a gene. A locus can only be occupied by one gene, but this gene can have different forms, and the alternate forms are called alleles. In the example of peas, we saw that the color-determining gene can have two alternate forms, big Y and small y. These are alleles, and as such, they will reside at the same locus on a chromosome. Chromosome pairs become separated during sexual reproduction, so that each sperm or egg cell produced has half of the normal set of genes. Sperm and egg cells are called gametes, but they are also called haploid cells because they contain half the number of genes and chromosomes. The process of producing cells with half the number of genes and chromosomes is called meiosis. Meiosis ensures that when sperm and ovum fuse to form a fertilized egg, called a zygote, the number of genes an offspring has is doubled and thus returned to normal. By this mechanism, two gametes fuse to form a zygote, so the two haploid cells fuse to form a diploid cell. When body cells divide, they double the number of chromosomes they contain just prior to forming two new cells, so that each new cell has the normal number of genes, a process known as mitosis. In order to comprehend the role that genes play in nature, it is important to understand what a gene is and the role that it plays in the creation of an organism. So, what is a gene and what is its relationship to a chromosome? A chromosome consists of a lengthy string of genes. A gene is a portion of DNA that, via its sequence of codons, codes for protein synthesis. And proteins, of course, are the main building blocks of the body and brain. But let's break it down a bit. Physically, it is a double strand of the chemical deoxyribonucleic acid, or DNA, the structure of which was discovered by Watson and Crick in 1953. DNA is a truly giant molecule, sometimes as long as 2 inches, and resides within the nucleus of the cell. In structure, the famous double helix DNA has the shape of a twisted ladder, with each of the rails, known as the backbone, being made up of alternating units of phosphoric acid and deoxyribose sugar. Each link of acid and sugar is connected to a base that may come in one of 4 forms. These bases are A, T, C, and G, with adenine being A, thymine T, cytosine C, and guanine G. Each link of sugar, acid, and base is called a nucleotide. The rungs of the ladder are made up of 2 paired bases. The properties of these bases are such that adenine can pair up with thymine and cytosine can pair up with guanine. Bases code for the production of amino acids, which are the building blocks of proteins. Each amino acid is coded for by a triplet, or codon of bases. The amino acid lysine, for example, is coded for by the codon AAG. We can think of each codon as a different word and a gene as a sentence made up of these words. It is the precise sequence of the 1 billion words that makes up human DNA. Recently, however, it has been discovered that the vast majority of our DNA does not code for protein formation, but that much of this non-coding DNA plays an important role in controlling the activity of the DNA, which does code for protein production. In fact, it is now known that genes which directly code for proteins make up a mere 2% of our DNA. These recent findings have led to debates about how we define what a gene is. We can think of each codon as a different word and a gene as a sentence made up of these words. It is the precise sequence of the 1 billion words that makes up human DNA. The use of 3-letter words from an alphabet of 4 letters leads to 64. 4 times 4 times 4 different possibilities. This is more than ample for amino acid productions, since only a little over 20 amino acids exist in nature. This means that a number of slightly different 3-letter words have the same meaning, like synonyms. For example, AAG and AAA both code for lysine. Although there are only around 20 different amino acids in existence, by stringing these together in various sequences, hundreds of thousands of different proteins may be created. So let's briefly review. A gene is a portion of DNA that, via its sequence of codons, codes for protein synthesis. And proteins, of course, are the main building blocks of the body and brain. Recently, however, it has been discovered that the vast majority of our DNA does not code for protein formation, but that much of this non-coding DNA plays an important role in controlling the activity of the DNA. Despite such recent discoveries and debates, the main function of DNA in all species is still considered to be the production of proteins. But how exactly does DNA do this? While this image is a bit more detailed than you need to remember, it may help clarify the process. When a specific protein is required by a cell, a portion of the double helix unzips, revealing a sequence of bases. Other free-floating bases within the cell attach to the exposed bases according to base pair matching, forming a second type of molecule, messenger ribonucleic acid, or mRNA for short. This mRNA, once formed, detaches and travels to cellular organs called ribosomes, where, via another form of RNA called transfer or tRNA, it forms a template for amino acid and hence protein synthesis. The formation of proteins from DNA is called translation. DNA has one other main function: to make copies of itself, otherwise known as replication. Replication occurs when a strand of DNA fully unzips to form 2 separate strands. As before, free-floating bases attach to the exposed bases on the strand to form 2 new identical strands. Each species may, in theory, be defined by the specific proteins that its DNA produces. Our own species has perhaps as many as 100,000 different proteins that make up the body and all of its internal chemical reactions. Given that 1/3 of all protein-coding genes are expressed only in the brain, meaning they only produce proteins in the brain, many behavioral biologists believe that differences in personality and intellectual ability may, in part, be caused by differences in the genetic code that we inherit. Let's review the structure and function of DNA in another way.
DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine. AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell.
Although a draft sequence of the human genome was originally published in February 2001, it was not until April 2003 that the entire human genome had been sequenced. Humans were not the first species to be sequenced, actually the 40th. The DNA for 39 species of bacteria had already been determined, as well as a species of yeast, a nematode worm, the Drosophila fruit fly, and a mustard weed. But unraveling our own DNA was arguably a major triumph of molecular biology. Some evolutionists have claimed that this is the biggest scientific development the world has ever seen. Given that our genes vary from person to person at hundreds of different loci, whose DNA has been sequenced? The Human Genome Project has actually produced the average or consensus human sequence for 200 people. What does knowing the human genome sequence really tell us about how we are built? Now we know the precise base pair sequence for human DNA, and as a consequence, we also know how many genes it takes to build a person. You might assume that knowing the base pair sequence allows us to determine the polypeptide sequence, and that this allows us to determine the precise number of proteins that make up a human. But this is not true. Many polypeptides can fold themselves up in different ways, resulting in different proteins. This is why, although the base pair sequence is now known, this information alone does not tell us the protein sequence. It also helps to explain why, although humans have perhaps 90,000 to 100,000 proteins, these are created from fewer than 22,000 genes. Which is not much more than the fruit fly with 13,000 genes or the nematode worm with 18,000. The results of the Human Genome Project should be seen as only the beginning of the understanding, and just a part of what it takes to build a human rather than the endpoint, as many have previously suggested. Animal breeders have long taken advantage of the emergence of individual differences in their livestock to breed for exaggerated features. The estimation of the extent to which we are able to breed for a characteristic is called its heritability. Evolutionists define heritability as the proportion of the observed phenotypic variance that can be attributed to correlated variance in genotypes. This is an estimation of the extent to which a trait in a population is due to genetic influences rather than environmental influences. Let's look at egg production to illustrate the notion of heritability. Note that estimations of heritability must always be qualified by specifying the population or, in effect, the gene pool. In a different population of hens, the heritability for these two characteristics might be equal or even reversed. In recent years, egg producers have been able to increase the size of domestic hen eggs far more successfully than they have been able to increase the number of eggs produced by each hen. This is because there is more genetic variability available for egg size than egg number. Egg size, therefore, has a larger heritability than egg number. In the red Burmese jungle fowl, the wild equivalent of domestic hens, the number of eggs laid is much smaller and varies with the seasons. Since jungle fowl have not been selectively bred to produce so many eggs, it may be possible to increase the number of eggs greatly. So for red Burmese jungle fowl, the number of eggs laid may be highly heritable when compared to their domestic cousins. Studying the heritability of egg production certainly has practical implications. An egg producer's livelihood may depend on it. But when we turn our attention to human cognitive and behavioral characteristics, the notion of a genetic basis for such features takes on social and political connotations. The idea of human abilities being largely inherited has been misused on a number of occasions during the 20th century. It is important that we are clear about the limitations of heritability studies. To say that a characteristic is estimated to have a high degree of heritability does not mean that little can be done to modify it by changing the environment. Adult height may be a highly heritable characteristic in humans, but height may be substantially reduced when a child is raised with a poor diet. So, how might we estimate the degree of heritability of human abilities? One method is to examine the phenotypic variation in people who are genetically related to different degrees. Such studies compare the correlation between monozygotic identical twins with the correlation between dizygotic non-identical twins on various traits. The idea here is that since identical twins share 100% of their genes, whereas non-identical twins share only 50% of theirs, we would expect the correlation for any trait between the former to be much higher than between the latter if genes play a prominent role in the trait. Studies of the correlation between IQ scores in twins, for example, suggest a correlation of around 0.75 for identical twins and around 0.38 for non-identical twins. This finding has been taken by behavioral geneticists as evidence that intelligence shows quite a high degree of heritability. We should bear in mind, however, that 0.75 is not a perfect correlation, thus leaving room for environmental influence. In contrast to intelligence, most studies of personality traits suggest about 40% of the variation is due to genes, and around 60% is due to environmental differences. Up until now, we have considered natural selection to be the cause of change in organisms over evolutionary timescales. Today, natural selection is certainly seen as the prime mover in evolution. However, other causes of change can occur in a population. When animals move from one population to another, they may, by chance, have a genetic makeup different from the new population. If the new makeup confers an advantage in the new local environment, then the population can alter quite rapidly. This process is called gene flow, since new genes flow into a new environment. Another force for change, called genetic drift, consists of changes in a population that may build up by chance because they are selected neither for nor against. Genetic drift may operate in all populations, but is believed to be a significant factor only in very small ones, since each chance meeting will have a bigger impact. Occasionally, however, genetic drift might have a major impact on a population due to a process called the founder effect. This means that when a new population is started by a small number of individuals, as might happen when a group of rodents colonizes an island for the first time, then they are likely to have only a small proportion of the genes from the population they left behind. In these circumstances, the founder effect may mean that genetic drift is initially of great importance. This means that under some conditions, processes other than natural selection can lead to evolutionary change. But under most circumstances, natural selection is the prime mover of evolution. Currently, two competing views exist as to where and when modern Homo sapiens emerged. The multi-regional hypothesis is the view that gradual changes in numerous Eurasian populations of Homo erectus led to anatomically modern humans. If this argument is correct, then our direct ancestors lived outside of Africa for well over 1 million years. In contrast, the out-of-Africa hypothesis suggests that a later African population of Homo erectus led to modern Homo sapiens, and that these then gradually spread out of Africa, displacing earlier hominids. If this argument is correct, then we all share a recent common ancestor that lived in Africa between 100,000 and 200,000 years ago. It also suggests that our ancestors have only recently colonized the rest of the world, recently on a geographical timescale, that is. The advocates of the multi-regional hypothesis rely largely on their interpretation of the fossil record. The proponents of the out-of-Africa theory also rely on an interpretation of the fossil record, but additionally they make use of another, more powerful tool, the molecular clock. Mitochondrial genes remain uncontaminated by sexual reproduction or selection pressures, and the only changes that occur in their DNA sequence arise through random mutations. Since the mutation rate is well known for mitochondria, it's somewhat higher than for nuclear DNA. Then, by comparing the variability of base pair sequences between people from different geographical regions, molecular geneticists have been able to estimate when our most recent common ancestor existed. It is these changes in mitochondrial DNA that constitute the molecular clock. And the figure that molecular geneticists have determined through this technique is around 150,000 years ago. The molecular clock is considered such a powerful tool because, whereas a fossil might have left descendants, all of our mitochondrial DNA certainly had ancestors. Since the mitochondrial DNA is only passed down via the female line, the earliest common human ancestor of present living humans has been called mitochondrial Eve, or sometimes African Eve. Hence, today, many evolutionists support the later out-of-Africa argument and claim that African Eve was indeed the common ancestor of us all.